Hi, welcome to my online presentation. I'm Bastien Le Gars Santoni. I'm a PhD student at the RMS Foundation, located in Betlar in Switzerland. And today I will talk about the effect of the calcium over phosphate molar ratio on the osteoclastic resorption of pure beta-TCP cylinders. So the main problem is if we have large bone defects because of a trauma, like this massive ski crash, or because of diseases such as tumor or bone cyst, the self-healing ability of bone will be not sufficient to repair such large defects, and consequently, we will need to use bone grafts. And especially among the synthetic bone graft, the calcium phosphate are really promising material because they have a very close chemical composition than the mineral part of bone. In particular, the one phase among the calcium phosphate, called the beta-3 calcium phosphate or beta-TCP, is very interesting because it has a cell-mediated resorption. Indeed, once beta-TCP will be implanted in the human body, it will interact with cells. And one type of cells, the bone resorbing cells, called the osteoclast, will be able to adhere on the surface of beta-TCP and locally secrete acidic condition in order to resorb the beta-TCP. This is illustrated here. So you can see here the path of the osteoclast. And if you zoom in with the scanning electron microscope here, you can see that the grain were resorbed. In particular, so here we have a simplified phase diagram, and beta-TCP has the particularity that it has a calcium over phosphate molar ratio of exactly 1.5. If this ratio is slightly above 1.5, we have beta-TCP, which is present with hydroxyapatite as a secondary phases. And otherwise, if the ratio is below 1.5, then beta-TCP coexists with beta-calcium pyrophosphate. Interestingly, the ISO standard actually specified that commercially beta-TCP can contain up to 5 weight percent of secondary phases such as hydroxyapatite or beta-calcium pyrophosphate. This means that commercially beta-TCP may have calcium over phosphate molar ratios ranging between 1.47 to 1.51. This leads us to the aims of our study, which are to assess the effect of small deviation from the 1.5 calcium over phosphate molar ratio of beta-TCP on the bulk properties of beta-TCP cylinders, so namely for the physical properties, their grain sizes and their relative densities, and for their biological properties, the osteoclastic resorptions. In addition, in this study, we also wanted to assess the influence of the sintering atmosphere on the bulk properties of beta-TCP cylinders. So again, for the physical properties, the grain sizes and the relative densities, but also now the surface chemistry. And for the biological properties, again, on the osteoclastic resorptions. To fabricate dense beta-TCP cylinders, first, we had to synthesize calcium-deficient hydroxyapatite powder via an aqueous precipitation method. Then we converted this powder into beta-TCP by a specific thermal treatment at 850 degrees for 1 hour. Then the powder was mixed with water and with Darvan, a dispersant, in order to make a slurry. And then with this slurry, we were able to pipette and eject this mixture in molds in order to make beta-TCP cylinders. This procedure here is called slip casting because underneath the molds, we have a gypsum base which permit to remove the water from the cylinder and make green bodies. So after that, so we can demold, we collect the green bodies, and then we have to sinter them. So we use the conventional sintering, so namely 3 hours at 1100 degrees. So basically, we just place the beta-TCP cylinders on a zirconia plate in the oven. Or we use a different setup, which is the sintering in the monetile-rich atmosphere. For this, we placed a crucible at the top of the cylinders, and we sealed the bottom here with a monetite powder. Of course, after the sintering, all these cylinders were polished. And part of the cylinders obtained with the conventional sintering were passivated or annealed at 500 degrees during 24 hours. So here is a summary of the factorial plan that we did for our study. So first factor is the cell culture type. So we had 3 levels. So one with cell culture medium alone, one with macrophages, and one with osteoclast. Today we'll only focus on the osteoclast. For the sintering, we have 3 different levels that we have already introduced. For the bulk calcium over phosphate molar ratio, we have 6 different levels, so 6 different batches with 6 different calcium over phosphate molar ratios, close to 1.5. And finally, we performed 3 repetitions for all these measurements. So concerning the physical responses, so the grain sizes were analyzed with a morphometric image analysis with the scanning electron microscope. The relative density of the cylinders was calculated with the Archimedes method in isopropanol. And finally, the chemistry at the surface of the cylinder was analyzed with XPS. For the biological responses, and namely the osteoclastic resorption, we choose to quantify the mean resorb thickness using a white line interferometry method. So just a few words about this method. So basically here we have our cylinders that was resorbed by the osteoclast, and we just take a picture of the surface which is resorbed. And then, so with the white light interferometry measurement, it's possible to extract the topography of this surface. And here you can see the color actually, which shows the topography, and green represent the reference plane, so the zero plane. And blue colors are actually places, areas that were below this reference plane. So in other words, places that were resorbed by the osteoclast. And then with the software, it was possible to calculate the mean resorb thickness from there. So concerning now the results, we can see the grain size as a function of the bulk calcium over phosphate molar ratio measured by X-ray diffraction. What we can see is that by increasing the calcium over phosphate molar ratio, we significantly reduce the size of the grains, and also that sintering in a monetite-rich atmosphere significantly reduces the size of the grains. Conversely, the relative density increased significantly with the bulk calcium over phosphate molar ratio, and the fact of sintering the cylinders in the moneta-rich atmosphere significantly increased the relative density of the cylinders. These two results are important because it suggests that the bulk calcium over phosphate molar ratio and the sintering atmosphere both influence the sinterability of the cylinders. And finally, we can see here the evolution of the calcium over phosphate molar ratio at the surface of the cylinders as a function of the bulk calcium over phosphate molar ratios. And what we can see is that annealed beta-TCP cylinders in green here had much higher calcium over phosphate molar ratio on their surfaces. And this is due to the fact that during sintering, we have evaporation of phosphorus, which led then to higher calcium phosphate ratios. Conversely, the fact of sintering cylinders in the monetite-rich atmosphere permitted to prevent this evaporation of phosphorus, and then we obtain surfaces with calcium over phosphate molar ratio significantly lower. As I said, so the grain boundaries plays an important role for the beta-TCP and its sintering and for its physical properties. And that's why we chose to send some cylinders to the team of Baptiste Gold at Max Planck Institute in Düsseldorf, so that they can analyze some of the grain boundaries in our cylinders. So from the cylinders, they cut a tiny microtip. And then this microtip, they were able to analyze it in atom probe tomography with a mass spectrometer. And here we can see the 3D reconstruction with all the elements that they have analyzed. And interestingly, in the center, we can see a line. And if now you draw the composition profile perpendicular to this line, which is actually the grain boundary, we can see that there, locally, we have an increase of the calcium and a decrease of phosphate and oxygen levels. And this is important because this suggests that the grain boundaries of beta-TCP have a different chemical composition than their bulk. And in addition, the fact that it's depleted in oxygen and phosphorus indicates also that the grain boundaries may be quite sensitive to the sintering atmosphere. And here are the results from the osteoclastic resorption. So you can see the evolution of the mean normalized resorb thickness as a function of the bulk calcium over phosphate molar ratio. So here we can see clearly a quadratic effect on which the resorption is maximum at calcium over phosphate molar ratio close to 1.5. And the fact of sintering now in the monetide-rich atmosphere significantly increased the osteoclastic resorption. This is illustrated here, while the fact of annealing the cylinders significantly reduced the osteoclastic resorption, and this is also shown here. So to sum up, the calcium over phosphate molar ratio and the sintering atmosphere modified the sinterability of beta-TCP cylinders and their osteoclastic resorption. The grain boundary composition was found to be different from the bulk composition of beta-TCP. And as outlook, so we will need to perform further characterization with the atom probe tomography in order to characterize the chemistry of the grain boundaries as a function of the bulk calcium over phosphate molar ratio of beta-TCP cylinders and as a function also of the sintering atmosphere. So I thank you for your attention, and if you have questions.